Hi, good morning, everyone, and thank you for joining me today for the first installment of the ACell webinar series. My name is Josephine Lembong. I am the Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. Today's webinar is on the topic of generation of QTPP, risk assessment, and CQA identification. So over the next hour, you will hear about these foundational concepts of quality by design and cell therapies from one of our lead contributors of ACell, who I will introduce in just a few minutes. Before we get started, I would like to provide a brief background on the ACell project to set the stage a little bit. ACell is a collaborative case study on the implementation of quality by design principles in cell-based therapies. This is a project that ARM undertook several years ago after feedback from our members about some of the CMC issues that the field as a whole is experiencing. It occurred to us and our members at that time that these issues were not unique to cell and gene therapy, and in fact, some other segments of the biologics industry had gone through similar growing pains before. And the example of the AMAP document was brought up. It was something that was instrumental and one of the things that allowed advances to occur in the monoclonal antibody industry in terms of improving manufacturing quality, optimization, and not to mention to drive down the COGS. So that was the impetus for ARM undertaking the ACell project. A lot of you might have known that more recently, we also published AGene, which is the sister project to ACell, and it's on the best practices on the manufacturing of AAV vectors, also using quality by design principles. So to make ACell an effective resource and reflective of the ongoing innovation in the sector, we chose to focus on immune cell therapy as a case study. This is based on strong performance and the field's enthusiasm of the cell-based immuno-oncology sector, as shown at that time of the writing, and the strong financing in the first half of 2021, and also major increase in industry sponsored trials worldwide. And to further maximize the utility of the document, we felt that it was necessary to focus on one specific use case. So we decided to further focus this document on CAR T-cell therapy, and in particular, an autologous approach, given the recent approvals and the number of products that are currently available to patients in the market. We are very fortunate at ARM to have a large number of subject matter experts from our member companies. There are many contributors to this project from more than 30 ARM member companies. And we would also like to thank Nimble, who generously provided funding for ACell, which allowed us to employ technical writers, without which we would not be able to complete the project. This document is intended to benefit the entire field, so it is available as open access on our website at this link here. So what is the purpose of ACell? We got feedback that there's a real need to establish best practices for process development in cell therapies. So ACell will educate on the basics of cell-based therapies, particularly CAR T manufacturing, but it will also inform on the methods used to design and quality. We think that this document will act as a tool for teams to communicate with each other. And as the industry is growing and many people are coming in rapidly into this field, there are a lot of experienced individuals from pharma who are entering the field who may lack the knowledge of the particular unique challenges of cell-based therapy manufacturing. So they may understand QBD, but not necessarily the specifics related to CAR Ts. And on the other hand, there might be cell therapy experts in the field who may never have the experience to scale up a product while also maintaining process controls. So QBD is a solution to this problem. We hope that this document will be a bridge between these groups, therefore a workforce development tool. And we hope that this document will be used by product developers, educators at universities, and we also have the aspirational goal that this will be used to help educate regulators. This is what the contents of the document look like. As you can see, it covers quite a big scope. Chapter 2 covers regulatory considerations, comparability, and standards implementation in cell therapies. Chapters 3 and 4 are really the foundational principles of the applications of QBD. 5 and 6 touches on the starting materials and ancillary materials considerations. And then we go to the manufacturing process descriptions, analytics, control strategy, and last but not least, the facility design considerations. ACell is not intended to be read front to back, although you are certainly welcome to do that, time permitting. Each chapter, to an extent, stands on its own, although there's a common theme running throughout. Today's talk will cover the foundations of QBD that are laid out in chapters 3 and 4. This webinar today is the first out of a 7-part webinar series for the ASOL educational rollout. There are 6 one-hour webinars scheduled roughly monthly until April next year, covering various chapters of the document, and it will culminate in a workshop in May or June next year. The schedules for the webinars will be updated and posted in this link down here. And for a housekeeping item, you can use the Q&A function in Zoom to ask the questions, and we'll try to get to as many of them as we can at the end of the talk. And with that, I would like to introduce our expert speaker for today's webinar. Pinar Cicalese has over 20 years experience in the industry and is currently at Immatics, leading the CMC commercial strategy and global patient supply for adoptive cell therapy programs for various solid cancers. Pinar has experience in process development, GMP manufacturing, strategy and supply of biopharmaceuticals and cell and gene therapies at various companies. Prior to Immatics, she has been responsible for global manufacturing and supply strategy in GSK cell and gene therapy platform. And prior to this, she led the manufacturing science and technology group in GSK, delivering many late-stage clinical and commercial biopharmaceutical assets to the patients. Pinar has a Ph.D. in chemical engineering and is also a Fulbright alumnus. Pinar, thank you again for taking the time out of your busy day to be here today, and I will turn it over to you to start your presentation.
Thank you, Josephine. And before I start my presentation, I want to thank Josephine and the rest of the ARM organization for spearheading the generation of this valuable document. And it's a very valuable resource, and I'm very happy to kick off the presentation, the webinar series that ARM is going to host, and I am inviting everybody to join to those webinar series. Let me start sharing my screen. All right, can you see my screen?
Yes, we can.
Thank you. As Josephine mentioned, today we are going to focus on the generation of quality target product profile and risk assessments that leads to identification of the quality attributes, the criticality of the quality attributes in this quality target product profile. We are going to start with the foundational concepts of quality by design, and then go through target product profile and quality target product profile, an example of QTPPs, some of the attributes, and how to translate these QTPPs to CQAs, the critical quality attributes. The risk assessment, there will be a risk assessment example, and then towards the end, we will go into elements of control strategy development, and again, very at the end, the life cycle approach, how to use these tools throughout the life cycle of a product. Again, there is many foundational concepts. There's many acronyms that you will hear, QBD, TPP, QTPP. We are going to, as I mentioned, talk about quality by design, target product profile, quality target product profile, critical quality attribute. We will not touch too much on critical process parameter, performance qualification, and continued process verification, but those will be again in the life cycle, just briefly touched upon. What is quality by design? I want to start with the definition by ICH Q8 revision 2, and then kind of dissect it into how it is related to these acronyms. But the definition by ICH Q8 of QBD is a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. So the predefined objectives in this case is your quality target product profile and the linkage to your critical quality attributes within that quality target product profile. Your product and process understanding is coming from your understanding of the linkages of the CQAs to critical material attributes, or the other way around, how the critical material attributes impact the CQAs, and similarly, how the critical process parameters impact the CQAs. And based on this relationship, you will be able to develop the control strategy, and the sound science and quality risk management comes into what you use throughout this development. In the right-hand side, you see a flow diagram, and the orange arrows are the risk assessments that you may have to do along the way. So this systematic approach is actually iterative. Throughout the life cycle, as you learn new things, you will be able to change your QTPPs, your CQAs, and your critical process parameters or critical material attributes. Some of the things may become critical, or some of the things may fall from critical, may not be critical anymore as you learn more. So this approach is risk-based, as I mentioned, throughout the process. Throughout the life cycle of a product, you will do many risk assessments, and therefore you are continuously improving your process. And QBD intention is to deliver a capable process and at the end, a quality product consistently. What are the differences between TPP and QTPP? Target product profile covers many aspects, more than what the quality target product profile may cover. It can describe the mechanism of action for a drug, clinical pharmacology, indications and primary and secondary efficacy endpoints. The intention is to use this profile as a vision, common vision for this drug product across all disciplines. So it can be used in commercial and business communications or net value calculations for the product. It also establishes basis for the drug development program, and it is linked to the strategy that you may want to use in clinical trials or in the non-clinical side of the business. Quality target product profile, on the other hand, is linked to CMC and is focused to characteristics of the product to ensure safety and efficacy. And let's take a look at the definition. Again, going back to ICH Q8, QTPP is defined as prospective summary of the quality characteristics of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the drug product. By focusing on these quality characteristics, the developers can focus resources and efforts on the most critical aspects of process. And in the right-hand side, you see it's a cyclical process. You define your QTPPs, then you identify the CQAs, and based on that, you can establish what type of process parameters are critical to impact these CQAs, and that is the foundation of your control strategy. As you learn more, you can redefine your QTPPs or CQAs and CPPs. So QTPP should capture entire body of knowledge about the product and the research experiences you may have about the product, available literature, relevant literature, some of the small-scale development activities, as well as the risk assessments or regulatory requirements and guidance. Let's take a look at an example. This is actually more of a categories example. And this is a typical content of QTPP. You will also find this table in the ACEL document. The first part of the QTPP here is related to the drug product design attributes. And the second part is the quality attributes, the attributes that are linked to safety, identity, strength, purity, potency of the product. Under the general properties, you can see therapeutic indication can be one. In this case, it was the relapsed or refractory diffuse large B-cell lymphoma or acute lymphoblastic leukemia. You can define your shelf life under this general properties, general attributes as well. What is your vision for the product? How long should it be stable? Storage conditions, and this can be storage conditions during transportation or storage conditions during the delivery setting as well. Container closure system can be listed under here as well, whether it's a bag or vial, whether it's sterile sealed or however you want to define your vision for the drug product. The dosage form, dose regimen, whether it's a daily, monthly, or single infusion, most of the allogeneic and most of the autologous products are focusing on a single dose, but there may be in the future, there may be deliveries that are repeated as well. Then delivery volume per dose, you can see, is it a certain dose or is it differing doses, differing volumes per kilogram of the patient? Then method of administration, most of these products are IV administration at this point. Then coming to general attributes, pH and osmolality, of course, these are related to the stability of the product and generally related to the delivery setting. If it's an IV, it needs to be probably at a certain pH and osmolality in order to not to create a undesired reaction in patient. The appearance is another quality attribute. Safety, usually microbial linkages, microbial testing and microbial attributes are listed under safety. For identity, we can say, for example, physical or chemical characteristics of the product. Going back, physical or chemical characteristics of the cell line, or it can be qPCR based, or it can be tissue specific gene expression, phenotype, genotype. You can, depending on your product, you can define what the test is most appropriate for you. And the content is total cell number, cell concentration, and active cells, transduced cells can be listed under content or strength. Purities can be divided into two. One, product-related impurities, which are live cells versus dead cells, so viability, or cellular impurities, or vector, etc., can be listed under process-related impurities, your residual media components and some formulation components can be under these impurities. Then potency, obviously, is the thing that you have to define, the test you have to define that is the most relevant for the biological function. And it can change from early stage to late stage in your clinical development. So during early stage, you may look into some specific markers whereas in later stage, you may want to look into more functional assays, and that may be expected. So additional considerations can be secondary indication or turnaround times, or you can also hear the vein-to-vein time for autologous products, patient weight range or age range, because certain patients may be beyond what you may want to treat, volume per cryobag or how many cryobags, maximum number of cryobags you may want to have, short-term storage conditions and transport conditions, as I mentioned before. Let's take a look at more details into the next slide. And this is an example of autologous cell therapy QTPP. Again, you can see this example in the ACell document. Here, the general attributes, as we talked about, are pH and osmolality, and these are mainly related to stability, and it needs to be compatible with the route of administration, which is IV in most of the cases, as I mentioned. Appearance, color or opalescence, in this case, the target is colorless to yellow, and visible foreign particles, in this case, target is negative. In some of the cases, there may be a certain number that you may want to specify here as well. The content and strength related attributes, the dose range, the cell concentration, you can see in this case CD3 plus transduced cells per weight is determined as the dose range, and then cell concentration is how many cells you have per ml. Product and container volume, in-use stability, so stable at room temperature so many minutes before infusion. So those may need to be determined. Those may need to have certain targets, again, depending on your product specification and product vulnerability to degradation at room temperature. Safety tests are usually coming from regulatory expectations. You have to have certain sterility, mycoplasma, endotoxin, and bacterial contamination testing. And you can see in these cases, sterility, mycoplasma, and endotoxin are compendial methods. The RCL and replication competent lentivirus and vector copy numbers are also under safety tests, because we don't want the lentivirus that we are using to transform the cells, to get our genetic load into the cells to start replicating in the patient's body. Then the identity in this case was, again, CD plus cells, and it's tested by flow cytometry. Let's take a look how these are linked to CQAs and what does it mean to define the CQAs. So again, I want to go back to the ICH Q8 definition of a CQA. It's a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. CQAs are related to the patient's safety and product efficacy. So therefore, CQAs are usually on the release specifications. So they will define your specifications, and they can also help in your product comparability as well. Throughout the life cycle of the product, in early stages, you may start with a certain process, and later on, as you go through different clinical trials, you may change your process. And in those cases, comparability is very important to be able to relate the clinical experience you have in the early phases to the later phases, especially for the products that are complex, like cell therapy products. And you can look at your CQAs and how your CQAs are changing, or the ranges of those CQAs are changing or not changing, and that can support your product comparability while you change your process, that the end product is not changing, and therefore it is comparable, and therefore the clinical data that you got with your earlier processes are relevant to your later processes because your product is the same. So again, this is the foundation for managing product quality through all product lifecycle. And CQAs obviously can be applicable for both drug substance and drug product in this case. For example, for vector manufacturing, it can be also certain CQAs that are defined. How do you translate QTPPs to CQAs? The QTPPs serves as a starting point to establish CQAs. And QTPPs lists all the attributes that are relevant to the product quality. However, not all these attributes may be critical. So the ultimate measure of criticality comes from risk assessments that you have to perform. And as I mentioned, these risk assessments are tools to combine, tools to get a summary of all the knowledge you have for the product. Not only the knowledge you have from as a developer from your clinical studies or preclinical studies or process development, but also you can gather relevant scientific publications and relevant research papers, relevant literature from other parties. And the formal risk assessment needs to be multidisciplinary because every group has a certain expertise, and they bring many different expertise onto the table, and you have to capture all these knowledge in your risk assessments to define the criticality. Otherwise, you may miss some things. The identification of CQA, however, does not indicate a test needs to be performed for that CQA, because some CQAs may be highly challenging. For example, stem cell engraftment can be one of these, so you may not be able to test it analytically. Therefore, you may accept the risk and look into clinical experience later on. Or CQA the testing the CQA may be unnecessary, since the manufacturing process design will always control the CQA. But that doesn't take away from the criticality of this attribute. As we mentioned before, CQAs can be revised as more process understanding is gained and more product understanding is gained as well. I want to go back and repeat the categories, the attributes that we have seen before. Safety, again, it can be certain general safety related, such as endotoxins, sterility, mycoplasma, or adventitious virus, or some product related ones like RCL or VCN. And identity, expression of surface markers, can be a general attribute. However, confirmation of genetic modification to your specific modification can be a product-related attribute. Then again, we talked about purity and impurities. There can be cellular process-related impurities, but you can also have untransduced cells or residual vector, etc., defined as your additional product-related attributes. Potency can be functional response or biological activity, and pH and osmolality we listed on under general attributes. Appearance, color, opalescence, foreign particles. These are, again, general attributes that are expected. And the content, total cell number, concentration, viability, and quantity of genetically modified cells can be some of these attributes that you may want to go through, take through risk assessment. So how do you define the criticality? And ICHQ9 defines the risk analysis as the qualitative or quantitative process of linking the likelihood of occurrence and severity of harms. So in this case, again, depending on the stage you are, you can look into severity and uncertainty of the information, or you can split severity into two. You can look at risk of impact to efficacy and risk of impact safety separately. But still, in these cases, you still have to look into uncertainty as you learn more and more about the product, you will reduce this uncertainty. Again, some safety attributes, as we talked about, like sterility, endotoxin, mycoplasma, etc., they can be obligatory, therefore may not need to be risk assessed. You can say these are already CQA, is expected by regulatory agencies, and it's safety related, so we don't need to rank these. And again, some of the uncertainty ratings may be theoretical, so you may want to capture this as a higher uncertainty or lower uncertainty depending on your knowledge. And let's take a look at these rankings, how to rank severity uncertainty, a little bit more detail. Severity, as we mentioned, refers to how significantly the attribute would impact the product's safety or efficacy. And let's take a look at low, for example. In this case, variability in attribute has minor or negligible potential for decreased safety and efficacy. So in this case, again, you can rank this attribute with just low, or you can give it a value depending on how you want to approach your risk assessment. And if the severity is high, this variability in this attribute may have a potential for severe effect on patient and potential significant change in efficacy, safety, or risk-benefit profile, and may result in a serious adverse effect. We are going to see an example soon. So before we go to the example, I want to show the uncertainty scoring. And uncertainty refers to the level of confidence in assessing the criticality of the attribute. Obviously, these cell and gene therapy product is a new class of therapy, and the literature and knowledge on these classes of therapy is much smaller when compared to some of the other types of drug products. Therefore, in some certain cases, some attributes we may think may have a high impact, may have a high uncertainty scoring as well. Let's take a look at these scoring. For example, for low uncertainty, your prior knowledge may be large, meaning well-characterized effect based on extensive in-house data or large body of knowledge in literature of similar class of products. Or you may have some preclinical study experience, or you may have significant clinical experience with this product. And for high uncertainty, you may have limited scientific understanding or no published data, and there may be no preclinical data or clinical data for this product. And in this case, how you determine whether an attribute has a CQA, potential CQA, or non-CQA is as follows. As you rank these attributes, a low and a low severity and low uncertainty ends as a non-CQA. In this case, low severity and medium uncertainty is also defined as non-CQA. In the high severity cases, whether you have low, medium, or high, everything is defined as CQA. This is one approach in defining your criticality. There may be other approaches as well. For example, for high severity and high uncertainty, you may define this as a potential CQA as well. And what this means is you still treat this CQA as, you still treat this potential CQA as a CQA and define and determine whether you need to get more information about this so that you need to so that you can reduce the uncertainty or you can reduce the severity linkage of this attribute. Let's take a look at some examples. Cell yield and viability can be an example attribute. And in this case, severity for cell yield and viability is high because if you don't have a certain yield or viability, for a certain yield, obviously, you may not be able to meet your dose and the efficacy of the product may be different or may change. And for viability, similarly, the non-viable cells may create an adverse reaction or it may decrease the efficacy. But in this case, uncertainty is ranked as medium. In any case, according to this table, we can rank cell yield and viability as a CQA. Of course, this can, depending on the process robustness, no action may be needed. And you may want to think about how your range, how your specification is defined for the CQA. If you set too tight of a specification on final cell yield or viability, it may pose a risk that product not meeting the specification. And different seeding cell densities, for example, can be implemented for this manufacturing process so that you can meet your viability or cell yield at the end. And of course, you have to still look into your other CQAs and whether, while you are changing this seeding cell density, whether you are also changing the profile of the cells that you are generating at the end of the production process. And another example is residual beads as a process impurity. And we know that residual beads could elicit some type of a unwanted effect, toxicological or pharmacological effect in the patient. And there is some literature evidence of that. Although you may not have evidence in your product, you may still be able to rank this attribute as high severity. And although there is no standard acceptance criteria for residual beads, there is a general understanding that greater than 5 log depletion is accepted as a proof of safety. And therefore, the uncertainty score, you can give it a medium since you don't have this exact acceptance criteria. So it is still this attribute, residual bead levels, can also be a CQA in this case. Here's an example that you can see in a cell publication as well. And you can see here most of the safety attributes are ranked as with severity of 10 and uncertainty with low score. and therefore they are all listed as CQAs. And again, some of the others here, most of the things here are CQAs or potential CQAs. For example, residual plasmid DNA. The severity score is a medium, and uncertainty score is low, and therefore it is ranked as a PCQA, potential CQA. So you may ask, what are the example non-CQAs in this case? So in this example, all the quality attributes that are listed are CQAs. However, for example, there can be product-related attributes that you may want to check, such as cellular or apheresis-related impurities, for example, monocytes or granulocytes, etc. And they may end up being non-CQAs because they may not contribute anything, or they may not risk the safety or efficacy, the safety of the patient or efficacy of the drug product. Okay, you understand your product, but it's also important to understand your process. CQAs are an important step for your product understanding, but not the last one, because you have to produce your product, and therefore you have to define how to control your process parameters to define your control strategy. Not only your process parameters, actually, but also your material attributes that are incoming from your raw materials. Again, this is a very inclusive graph. Your product understanding is where you start. However, you have to get more process understanding and establish your control strategy along the way. And part of this establishing control strategy is also exploring your design space, which is normal operating ranges and proven acceptable ranges, NORs and PARs. And these are, you will see in the life cycle, comes later on in the life cycle. The clinical experience is also important because at the end, the drug product needs to be efficacious and safe for the patient. So therefore, you have to feed your clinical experience back to your product understanding and feeding into the process understanding. So again, it's a cyclical process, and it's an iterative process throughout the life cycle. Let's take a look at the life cycle. Most of us are familiar with the clinical and regulatory pathway. Like there's early product development, preclinical, phase 1, phase 2, phase 3, and phase 1 and phase 2 in cell and gene therapies may also be referred as pilots, and phase 3 may be referred as pivotal. And then comes your regulatory submissions, and obviously you launch your drug, and you have your post-marketing commitments. The early phases of this development, preclinical phase 1 and phase 2, or preclinical and pilot, can be referred to as process design phase. And as you come to pivotal stage, phase 3, you should be at process qualification. And before you submit your regulatory documents to regulatory agencies for product launch, product approval, you have to complete your process qualification. And then the expectation is you are not done, you are still learning as you manufacture the product and get more data from the patients. You will continue with your continued process verification. Let's see how these documents and the concepts fit into these stages of process validation life cycle. At the beginning, for stage 1, for process design phase, you have to define your QTPPs and CQAs and understand your CPPs, CMAs, NORs, and PARs, and establish your control strategy. Then later on, you will have a process validation master plan and your process performance qualification, protocols and reports. And at the end, after you launch, you have to continue your process, you continuously verify your process. You will have periodic reports such as annual product reviews, APRs, and updates to the lifecycle documents. Before I close, I wanted to reiterate certain concepts or certain important points. It is important to establish the QTPP early during your development because this can focus your efforts in your development and clinical study designs. The next step is obviously CQA identification, and CQA identification, this dictates the rest of product development and validation lifecycle. Again, as we visited, a risk-based approach is required and expected, but there is no one-size-fits-all, so you can define how you want to capture and how you want to rank. As long as you are consistent with your risk-based approach, you can tweak certain criticality levels according to your process and product understanding. Of course, I've also mentioned it's a cross-functional team of subject matter experts that are required because the criticality can be perceived as differently by these different subject matter experts. It's important to bring everybody together to be able to define the CQAs, the critical quality attributes. And of course, it is important to understand your CQAs and the linkages between CQAs, your CPPs, and clinical experience. And that's fundamental for your effective control strategy. At the end, we all work for patients. We all want to deliver quality product to the patients. And therefore, an integrated and iterative strategy it needs to be incorporated to the product lifecycle. And as you learn from your developments and clinical experience, you may need to go and revisit these criticality assessments. I have few links here that are useful for references. The ICH guidelines, I'm sure many of you are very familiar with them. There is the process validation guide by FDA, and there is another FDA guideline, draft guidance referenced here. And PDA technical report 81 is also very useful in defining how these processes, how to define the criticality and how everything will work together and how the iterations may need to be done. And with that, thank you very much. And maybe we can take some questions, Josephine.
Great. Thank you so much, Pinar, for the fantastic presentation. So we do have time for audience Q&A right now. Maybe as we allow the audience to type in their questions, I will start with one, Pinar. So ASOL was written, focusing on autologous CAR-T, and it's primarily written with a hematological cancer indication in mind. Could you comment on how this document can be applied for allogeneic products, or even broader for other indications or other modalities?
So most of the attributes we talked about will be applicable to all the indications. It's platform-based, cell therapy platform, and it's also applicable for autologous versus allogeneic. However, autologous is different from allogeneic. We all know autologous starts with the patient. We take patient cells transferred to our manufacturing centers, and we modify them and take it back to the patient. So in the case of allogeneic, your starting material is different. It's starting with a donor material. And of course, this changes the response or risk of certain things. For example, there is some risk of immune response or risk of graft versus host disease can be different. And for allogeneic products, you may want to consider those attributes as well when you are looking into criticality for the attributes, list of attributes.
Okay, great, Pinar. And to follow up on that, there's actually an audience question that just came in. What about cell-derived products such as exosomes? How do you think this is applicable to those as well?
I think the concepts are very applicable to all the products. I have a lot of experience with biofarms, and the same concepts apply to biofarms as well. Obviously, certain things are not applicable. The cell type, for example, or cell panel, different types of cells, the distribution of cells may not be applicable for non-cell-based products. However, looking at your product and defining the attributes that may define your product vision, and then going through the criticality assessment. So the foundations of the QBD is still applicable, whether this is a cell-based therapy or vector or a protein-based therapy like biofarms.
Mm-hmm. Okay, great. Let's see. I see there's a few questions that are on the timing about the risk assessment. So I think I'm going to group these all together. So, how early should you apply QBD, and should you start CQA risk assessment? And how do you do that in a very early stage when you lack process knowledge?
That is a very good question. The timing also puzzles a lot of developers. But to me, this risk assessment is culmination of everything, meaning you may be early in your development, therefore you may not have any clinical experience. Maybe early in your process development, and therefore you may not have any process understanding. However, you will have a vision of how your product should behave or should have an end result in the patient. And depending on this, I'm sure there's many theoretical things that you can find in the literature that can define these criticalities. There is really no one size fits all. My recommendation, since this is a summary of everything you want to have in the product, as well as all the information you can gather from others' experiences, your experiences, and clinical experiences, it is very beneficial to start defining the attributes and start defining the criticality on these attributes. This doesn't mean those you define as critical is going to stay critical. You can state if there is uncertainties in the criticality and in this knowledge, you can define them as potential. But the sooner you start putting these knowledge together, the sooner you can unify all the experts you have in your company around this vision for the product. There's, again, no one size fits all. My recommendation is during again, the early the better, but definitely during stage 1, meaning during pilot clinical trials, to have this knowledge captured.
Okay, great. Thanks, Pinar. We have a couple of questions about product intermediates as well. So does a QTPP have to be defined, and purity or potency have to be established for intermediates like PSCs? There's another question also about the vector, especially for CAR-T.
Yes. So again, there's no one size fits all. However, yes, the critical quality attribute concept also is applicable to drug substance or, again, vector in this case. We may define as drug substance. And also the criticality is, again, this is a summary of all the knowledge. Therefore, it can be defined at any stage, meaning if there are critical attributes that may lead to drug product attribute impact, you can define these as critical attributes in your process. And of course, then what it leads to is, if it's an intermediate, you may want to try to control these parts, or you may want to limit what can enter into your process in case of critical material attributes, for example.
Got it. Okay. We have a couple questions on the regulatory side as well. So if you decide not to test a CQA because it is always controlled during the manufacturing process, what justification, if any, do you need to have in place for the regulatory submission?
Again, certain CQAs you may not be able to get away with. For example, sterility or certain obligatory ones. You may not be able to, even if your process controls at all times, you may not be able to change that. You may have to test for this. But for certain others, you have to gather your process information. You have to gather all the justification, such as, in X number of manufacturings we have, and you can include certain types of manufacturing that is not for clinical, for example, that can be also in process development, as long as the manufacturing that you are doing is comparable, meaning it's in a controlled environment. Certain inputs are controlled, outputs are recorded in the same way. You can gather all the information you have and make your justification to regulatory agencies.
Okay. We have 1 minute left, so we'll have time for just one more question. So as the QTPP evolves, at what point do changes in your process or your product attribute result in a new product?
That's a very good question. It all depends on how you justify your I guess changes. Meaning, do they change? Dropping a CQA to a non-critical level doesn't change your product, as long as the attributes in your product or the efficacy and safety of your product stays the same. So again, the comparability part comes into this, because throughout your process lifecycle, product lifecycle, you will change your process many times. But at the end, if you are changing certain attributes, let's say it's a totally different vector that you are using for a totally different target, not necessarily for, again, there's examples, NYSO or PRAME, etc. Those changing your target may change your product, but if your target is the same, if you drop a CQA, or even if you add certain things to your target, enhance your target, for example, it may not change your product. It's still under the same umbrella of the same product family.
Okay, excellent. So we are at time. Thank you very much, Pinar, again, for the fantastic presentation and for the Q&A. Thank you, the audience, for participating in the Q&A. We didn't get to a lot of the questions, so what we'll do is we'll try to get back to you offline after this webinar to answer your questions. So with that, I would like to wrap up this webinar today. Thank you again, and stay tuned for the next, the second webinar coming up on November 9th on the manufacturing. Have a great rest of the day, everyone.
Thank you. I can't wait to attend that webinar too.
Excellent. Thank you, Pinar. All right. Bye-bye, everyone.